of what David says. I hope good luck to the rest of you.
You win. Hopefully you win.
First nasal spray, steroid spray we had 40 years ago. Nasal beclomethasone.
So it's here. Is that okay?
Bacon.
Okay.
You get credit for both spellings.
Okay. It won't be the last typo, I promise you.
So right here, click share. No,
Oh.
it's just that. There you go.
Okay.
All right.
So you're sharing. Yep. Okay, so I hope to keep it simple, but I want to talk about gene editing or the basics of gene editing. And the reason is that if you go through downtown, just in front of Building 1, the Seattle Children Research Institute, you can see these neon signs. One of them says gene repair, and I always thought that it was interesting, and I didn't really know what it means at first. So I thought I should do some reading and explain to you what it means. Disclosure, so no disclosure. You promised new PIDDs the last time, so there will be no new PIDDs. And yes, I remember last year, so I really hope that 10% of you will survive the talk, and it's meaning 1.5 of the people in the room, which will be a great improvement from last year, and there will be no jokes this time because it's a serious topic. Seriously. Okay, so what are we going to talk about? So I'm going to talk about the general idea of gene editing or gene repair or gene therapy. We're going to talk about classic, which means old genome modification technologies, and in general we can think of them as a non-targeted gene transfer, and I will explain it later. And then we can we're going to describe new techniques for gene editing, which means targeted modification of genes. And for that, we'll go over several concepts. Designer nucleases, it sounds very fancy. Perfect. I think they have. Did I touch anything? Yeah.
We can listen to David.
So this is one of the achievements. Okay. So we're going to talk about designer nucleases, DNA double-strand breaks and how the cell repairs them, template delivery, and I will explain these later on, and then we'll end up by 1.5 examples and ideas which you should provide. So what is genome modification technologies, or what are we talking about? The whole idea about gene therapy, gene modification, is to take some DNA or nucleic acid and put it in a different place within the cell. So we hope to achieve chromosomal integration of edited DNA, and we hope it to be constantly or ongoingly expressed in the cells even after it divides, so it will be there. In gene therapy, what we hope to do is to take a gene, usually replace or add a gene which is missing inside the cell, and express it in the same cell or in specific cell population that we're interested in. And it can be done in targeted or non-targeted genetic manipulation, which means that it can either be done randomly, so we insert this nucleic acid randomly in the cell, or we have ways, designed different ways to put it exactly where we want to put it. What we need for that, we need a vector, a way to insert the DNA, to transfer it into the cell. We need some DNA templates, not all the time, and we'll get to that. And we need some nucleases to cut the DNA, which is also optional because sometimes you can do it without any nucleases. And that will bring me to the next topic, which is the classic modification technologies or gene transfer. And these are the initial studies that were done with gene therapy. So obviously viruses want to proliferate, and in order to do that, they infect different cells, transfer their own genetic material inside the cell, incorporate their DNA in the host cell DNA, and then they start to proliferate, making new viruses and spread themselves to the cells around them. So that's something the viruses can do. So people thought, okay, let's use viruses. They know how to do it. Let's change their DNA in some way and use them to transfer any DNA or nucleic acid material that we want to do it into different cells, human cells, for example, or any other cells that you want to do it. And the early approaches used gamma retrovirus to integrate viral DNA into the chromosome. There is a short scheme of the viral DNA, which is usually very simple DNA. It's a long stretch of DNA, and most of the viruses don't want to spend time or energy making sophisticated genes like we have in our cells with exon, introns. They just make a long run of RNA, and then they cut it later on, different proteins. So you have GAG protein, which makes the core proteins to wrap the viral DNA. You have the polymerase, which replicate the DNA, or other enzymes that help the viral DNA to incorporate into viral genome, to incorporate into the host cells, and then you have genes that makes the envelope. So people said, okay, we don't need all that. We don't need that in the virus. Let's cut those out and put in the genes that we want, and then just transfect any cells that we want to transfect or transduce. That's the right term. And these are, I brought two examples. So the first one was the XKID. So in XKID, you don't have the IL-2 receptor. It's IL-2 receptor deficiency, so you get a T-negative, NK-negative, B-positive cells. And this was an early study from 2002. They used gamma retrovirus to deliver the common gamma chain into CD34 cells, and that was done in autologous cells, meaning that they took the cells from the patient, repaired them, and just transferred them back to the patient. We'll wait with the update. Sorry. Yeah. Okay, so did it work? Yes, it worked perfectly fine, because in these 4 patients, one of them rejected the graft or the CD34s and had disseminated the infection at the time, so we can leave him aside. But within these 4 patients, you can see that they start expressing CD3 or T cells again. One of them even developed a thymus, which they don't have, so everything worked very nicely. Some of them even made antibodies, which is difficult to do. Sometimes you keep the patient on replacement therapy for a long time because it takes time for the B cell to get the T cell signal and start making antibodies. So everything was fine. But then a year later, came up the first report. So there was a serious adverse event in one of the patients, which means developed clonality and later T cell leukemia. And then several years later, 25% of them developed leukemia. So that was kind of disappointing. A second example, again, correction of X-linked CGD, using the same idea of taking gamma retroviruses, infect CD34 cells, and transferring them back to the patient. So they took peripheral blood CD34 cells from 2 patients. They were adult patients, transduced them, and then reinfused them 5 days later. And did it work? I miss anything? Yes, it did. So you can see, if you check the oxidized positive cells in the 2 patients, then you have positive cells. If we do the DHR test, when we activate neutrophil and see if they can oxidize the DHR, then you can get positive cells, and that's how you test for CGD. So obviously some of the cells were repaired, and it looked promising. But then a year later, and that's obviously I think 2 years later, another publication showing that one of the patients developed monosomy and myelodysplastic syndrome. The other one just lost his function. There was another problem of shutdown of the viral elements, so it stopped expressing the gene that was transferred. So we have problems with this approach, and gamma retroviruses cause malignancy, serious adverse effect. Why does it happen? Because this is something that retroviruses know to do. That's their whole purpose. So they have, obviously the virus want the advantage of proliferating and spreading itself. So retroviruses especially like open reading frames, or they like to incorporate inside active transcription site. So they don't just incorporate in different areas of the DNA. They want to get integrated into very active sites so they can replicate themselves. And they have another thing that calls LTR, long terminal repeats, which is a very strong promoter. So from the virus perspective, I not only want to get integrated in active sites, I also want to proliferate myself very strongly. So they're adding another enhancer. So they go into active sites, and then they're very actively replicate themselves to make more viruses and spread themselves. And if you take 1 and 2 together, so integration into proto-oncogene and strong expression of proto-oncogene results in malignancy. One of the sites that they were mostly found in is the limb domain, only 2, LMO2, something that people know today to look for and see if retroviruses or viruses get incorporated in. So what can we do today? Was that the end of the vectors? No, it was not the end of the retroviral vectors. People started to manipulate the virus. So they're using what we call today self-inactivating viruses or SIN vectors, which means that we remove these strong enhancer, these LTR, were removed and replaced with more physiological promoters. And people are also using insulators, something that blocks the viral DNA at the end, so it won't affect other genes around it. So that's an example. If you have a promoter, you have also sometimes enhancer that make the promoter works even better, and then if you add an enhancer, then you block the effect of the enhancer. So if the virus promotes replication of some genes, they add something else to block it. The typical one is beta-chicken globin. If you read about it, that's a typical enhancer. And they're adding another safety mechanism, like suicide gene. So you can treat the patient with some drug and kill all the cells that were treated. So in 5 years from now, when you get your gene therapy patient in clinic and he has some viral infection, you should ask him, you should know exactly what he was treated with, because if you're going to give him, for example, ganciclovir, you're going to kill all his cells. So that's something that we'll have to deal with in the future. And they use also other viruses, like lentiviruses. And lentiviruses, in contrast to retroviruses, integrate into genes, but not upstream, so not at the beginning of the gene, that's just in the middle of the gene. So if a lentivirus gets incorporated within the DNA, it will basically destroy the gene that it was incorporated in. So if it was an oncogene, for example, you won't get any activation of the oncogene. So the gene that you inserted will be in that site, in the dangerous site, but it will destroy the gene itself. So that's another safety mechanism. And as you can see, it wasn't the end of it. It wasn't even the end of the gamma retroviruses, because that was published in October of 2014. And again, people are using gamma retroviruses for X-linked SCID. And what they did this time is, again, as I explained, they developed self-inactivating gamma retrovirus vector. So they removed the long terminal repeat enhancer, and then they inserted, again, the IL-2 receptor into the CD34 cells and were able to cure the patient. Again, numbers showing that the patient are making CD3 cells, CD4 cells, CD8 cells, so everything looks okay. And this time they knew about the severe events, so they were looking specifically for integration of the virus in the susceptible loci. So this is, for example, the LMO2. In green, you can see integration of the old virus. So you can see many green dots, which means that the virus was integrated in many places. In many places in the promoter region, and in orange you can see the new virus, which still there but not as frequent. So is that safe enough? We don't know yet, but it might be safer. In any case, even if we do it perfectly, in a perfectly safe way, we still have some problems. So usually when you use a viral vector, your gene get inserted into random places. You can control exactly where it will get inserted. So what you get is insertional mutagenesis. So mutation, just because you put some nucleic acids in some random place. So you can target different genes, you can lose function of some gene, and who knows what can happen. And there is other thing that we forget. So IL-2 receptor is very nice. You need it in almost any cell to be expressed, so they proliferate, so they will proliferate. And you probably need it most of the time. But what happens if you need certain genes to be expressed only at a certain time? If you need it to be regulated, you don't want it all the time. So if you just take the virus and you infect cells, then you're going to get constitutive expression, which is not always a good thing, and I'll show you later on. And what happens if you don't have a missing gene? So you have a mutation and you have abnormal gene. And think about it, if they have to make dimers, so you get one copy of the normal gene, one copy of the abnormal gene, we know from hyper IgE patient that if you take one normal copy and one abnormal copy, you get a disease. So sometimes if you have to combine, your protein has to make dimers or trimers or multimers, then when you have one abnormal copy and you still keep it because you didn't do anything with that, then you're still going to have the disease and you didn't solve anything. Silencing of viral elements is another problem, but we can think about it later. So the new approach is to do targeted gene modification. You can call it gene editing. So instead of just randomly inserting a sequence, you modify a specific genomic sequence in living cell, hoping to change the function of the cell. So in order to avoid random integration, you need to use sequence-specific nucleases or designer nucleases because you want to break the DNA in specific point. And then you have to deliver your DNA template. And you have to count on intrinsic DNA repair mechanism to finish the job, and I will explain, I'll show you what it means. Some people call it genome surgery. I mean, if you have pediatric surgeon and you're doing an utero surgery, so I think genome surgery is the next. What do we mean by designer nucleases? Nucleases cut the DNA. So it means that we want to design a nucleases and tell it exactly where we want it to cut the DNA. And there are several types of, the more common designer nucleases are zinc finger nucleases, transcription activator like effector nucleases or TALENs, and that's the new kid in the block. It's the CRISPR-Cas9 system. So it's very common. I think almost in any paper that you can find or read about the CRISPR-Cas9, because they are very common now. What are the zinc finger nucleases? So zinc finger nucleases are fusion protein, the fusion of two components. One of them is the site-specific DNA binding domain, which direct the zinc finger to the right place. And the other one is an endonuclease domain, something to cut the DNA. So the specific DNA binding domain was adapted from zinc finger transcription factor. There are many transcription factors which use zinc finger to recognize specific sites or specific sequences in the DNA. And it composed of several domains. So each domain consists of about 30 amino acids. It has a conserved structure of beta sheet, these are the arrow, and alpha helix. And people learned that within, everything is stabilized by a zinc atom, so that's why it's called the zinc finger. And within each domain, there are several amino acids on the surface of this alpha helix, which recognize either 3 or 4 base pair DNA sequence. So it recognizes 3 or 4 nucleotides. And if you bind them together, so you can create a sequence or a tandem domain, you can create several fingers joined to each other, and then will recognize a longer sequence. So if you take 3 fingers, for example, they will recognize between 9 to 12 nucleotides. What is the endonuclease domain? It's usually a bacterial restriction enzyme. FOCI is something that's used frequently. And it's a weak restriction enzyme, which means that it only works as a dimer. It needs a helper from the other side to cut the DNA. What we get at the end is site-specific double DNA strand break. So we break the DNA. And how it works, so you have these zinc fingers. And if you link them together, here, for example, you get 4 domains of 4 zinc finger joined together. And each of them recognizes 3 nucleotides, and together you get a specific sequence which recognizes a unique sequence of 12 nucleotides. Now, the fact that this FOC, the cleave domain, is very weak requires another one on the other side. And people actually were looking for that because if you need 2, then you need another specific sequence on the other side, which make your targets very specific. Because you don't, I mean, it's like if you use primers to amplify some genes in a PCR reaction, usually the length is between 20 or 23 nucleotides. So if you think about it, if you select a length of between 20 or 30 nucleotides, the likelihood that you'll find the same sequence in a different place in the DNA is low. So you get a very specific site to cut the DNA. You take individual zinc finger, you bind them together, and that's another way to show it. You can specifically cut the DNA exactly where you want to do it. Is that clear? The concept? Okay. So talons, transcription activator-like effector nucleases, we call them talons. And tails are secreted by bacteria, usually plant pathogens. And what they do is to secrete these tails into the plant cell and convince the cell, these are actually basically transcription factor, they tell the cell, the plant cell, what to do, or secrete different proteins that will help the bacteria to survive or kill the plant. So basically it's telling other cell what to do. And that's a nice way to survive or promote your growth. And there are naturally occurring repeats. Usually these are repeats of similar structure of domain. And each domain also recognizes specific DNA nucleotides. So in this case, it's not 3 nucleotides. Each part of the talon recognizes specific nucleic acid, 1 nucleic acid. Each repeat is usually 33 to 35 amino acid in length. And in each repeat, there are 2 most important amino acids which recognizes a specific nucleotide. They're called repeat variable D residue, or RVDs in short. When people figure out how the RVDs work, they're able to change just 2 amino acids in each domain. And then you can combine them together and you can make a very specific sequence that you want to target. Combine it again with the foci, the same restriction enzyme, and you get a talon. The end result of that is that you again create site-specific double-stranded DNA break. Again, you break the DNA. How it works, similar to the zinc finger nucleotide. So we have this domain. Each domain recognizes 1 specific nucleotide, and you can change the just 2 amino acids. So NG recognizes specific nucleotide. Each combination of 2 amino acids will recognize a specific nucleotide. And then you can make a long run of these domains and make it very specific, make the cut very specific. What's the difference between talons and zinc finger nucleotides? Talons are easier to design, and if you read, you can order them online and get them 2 days later. And people are making libraries of talons, so you can basically target every site or every gene that you want. You don't have to design them now, and there are different companies that are doing that. You can just go online, ask what you want to do, or what you want to get, and you can target it. It's more complicated than that. If you want to do something more specific, you can always design your own talents to target specific site within the gene, but that's another issue. If you want to do it or order it online, it's available. Usually with talents, you get less frequent off-site targeting. So the major concern with all these designer nucleotides nucleases is that you're going to target some different sites. So you're going to make it break in a different place. That's something that is always of a concern. They're less toxic to the cells, but they are much larger. So if you need to put them inside the cell, you need to get them into the cell to break the DNA, it's going to be a problem. CRISPR-Cas9, it's a new system. So just to give you a general idea of the time, so the zinc finger are known for many years. I think it's more than 30 years. The talons became more familiar around 2006, 2007, and CRISPR-Cas9 system was described in 2012, and it's all over the place. People are using it everywhere now. So CRISPR is a clustered regulatory interspace short palindromic repeats, CRISPR, and it's part of an immune system, but it's a bacterial immune system. So what the bacteria are doing, they always can be infected by phage or other viruses, same as we do. And if a bacteria go to the doctor, doesn't feel well, tell me you only have a viral disease. Anyway, the bacteria collect sensing DNA segments, foreign DNA segments within them, and then they incorporate it into their genome in order to get rid of that. So how it works. So this is an example for how the CRISPR-Cas9 system works. So they have the CRISPR locus, and within this CRISPR there are repeats and there are spacers. So when a bacteria gets infected by a virus, it cuts the DNA just randomly. Any foreign DNA gets cut, and then it takes part of this foreign DNA and incorporates it into the CRISPR site. And now we call it a spacer because it replaced the spacer that was there. And then we'll focus on the middle. Then once this being activated, you get short sequences of complementary RNA, because when you transcribe the DNA, you get an RNA. And this short fragment of RNA goes to other places in which the foreign DNA got incorporated. So the bacteriophage doesn't stay only in one place. It goes randomly to different places inside the bacteria. So now the bacteria developed a way to recognize where foreign DNA got incorporated into her DNA. And the bacteria doesn't want it there, so it's going to cut that. And that's where the Cas9, which is another nucleases, get into the picture, and it cuts the DNA. Another way to show it, so you have the bacteriophage. It's inserting its nucleic acid. The bacteria cuts it, incorporates it into the Cas9. And then you get these short fragments of RNA which are complementary to the phage nucleic acid or genome, and it can recognize. And once you get this recognized, then they use the Cas9 nuclease to cut those foreign DNAs. Same way to show it, so you get this complementary sequence which the bacteria was able to make, and then there's a nick of RNA, and that recruits the nucleases to cut the DNA. So people said, okay, that's a very nice system. Let's use it for our purposes. Let's design our own guided RNA, a short sequence of RNA, and that will be complementary to some genes that we want to target, and we'll have the same system, and that will cut it. Then we'll cut the DNA at the exact place that we want to cut it. So the end result is that you can target or target the system to a different place that you want. Basically, you have an RNA-guided DNA endonucleases. And again, the end result is that you break the DNA. With all these three methods, the end result is DNA break. Is it good? Maybe it's good, but DNA breaks are not that rare. It happens in any cell many times through the day. So this is one of the estimated numbers. It's estimated that about 10 double-strand breaks happens in each cell every day. I read some much higher numbers. So this is a serious threat to the cell because it can kill the cell or induce genome instability. So cells has machinery to cope with that. And it happens all the time. It happens with exposure to DNA damaging agent, just UV light. Obviously with cancer, alkylating agent can do that. It happens sometimes deliberately. If you want to rearrange certain segments of the DNA, like in B cell, T cell, you have the VDJ recombination, and you're doing class switching recombination in B cells. So it happens all the time, these DNA breaks. And sometimes it's only the physical stress when cells are dividing, they're just pulling the chromosome to both sides, and then they can break. So the cells developed a very complex system to sense those breaks, because you don't want to divide or replicate a cell with a broken DNA. So the first thing they do is recognize that there is a break, and then they tell the cell to stop proliferate, arrest the cell cycle, because you don't want to replicate that. And they're just going on, and they fix the break. So we spend a lot of time to explain what different nucleases we have and how we can make the double-strand break, but the cell already knows how to fix it. So basically, I was wasting your time. But the truth is that we actually use. Sorry, I still have a lot to go through. So the truth is that we can actually use those breaks, and in general, without going deeply into the repair mechanism, there are two ways to repair a DNA break. So one of them is called NHEJ, that's a non-homologous DNA end joining, and you can see that there are many enzymes and proteins involved in that. Some of them should be very familiar: DNA, PK, C, ligase 4, Artemis. They are all causing skid, because if you can't repair the DNA, especially with those B cell and T cell that creating these breaks all the time, if you can't repair them, then you get a skid phenotype. And this can happen through the entire cell cycle. But there is a more sophisticated mechanism, and it's called homology-directed repair, or HR, which uses a template, and that happens only during the late S or G2 phase. Very clear, I'm sure. So non-homologous end joining, which means we got this break, so we have a gap within the DNA, and now this is an error-prone pathway. The repair mechanism just fills the gap, and it won't do it always perfectly. So sometimes it will do it perfectly fine, and you will get the wild-type cell, but sometimes it doesn't do it very well, so you get either frame shift because you didn't insert the right number of nucleotide, or you get insertion because you inserted more nucleotide than the break, or deletions because it's smaller. So in general, we can call them indels, insertions and deletions, and that changes the DNA. Is it good? Sometimes it's very good, especially if you think about T cell and B cell rearrangement. You want to make a big repertoire of TCR and B cell receptors. So that's one way to increase your repertoire, just random insertion and deletion. You change the sequence, you get a new B cell receptor, a new T cell receptor, everything is okay. In any case, now you don't have the broken DNA anymore. So that can happen during the entire cell cycle, because whenever you get a break, you can use this mechanism. You don't need any template to repair the DNA, but we can use it, so it's actually good for us. So if you think about it, you can knock out the target gene. If there is a gene that you want to study and you want to knock out it from the cell, then you can induce these double-strand breaks, and at least some of them will be repaired not in the right way, so you're going to lose your gene of interest. So that's a way to knock out some genes. What about homologous recombination? This is an error-free pathway. That's a good way, or we can think about it as a better way to repair deletion or DNA breaks. So for this mechanism, the pathway to happen, the cell is using another DNA template. So it needs another copy of itself to complete the break. So it can happen when you have two copies of each DNA or during DNA replication. So that's why it can happen during the S phase or the G2 phase when you have two chromatids. So it uses a sister chromatid as a template to correct itself. And then you get a very error-free repair, because you have the right template, you can complete whatever missing DNA that was, or nucleotide that were there, and you fix the break. But that also provides a perfect opportunity for us if you want to change the DNA, because you can insert your own template at that time. So instead of using the sister chromatid, you insert another template, your own template, and then you can change the DNA any way you want it. So the first step was to create the break, and then you can let the cell repair itself, but you can help it with the repair. So you can provide it your own template, and then you can get different end result or different end product. There is another problem. So you need to insert the template. You need to insert the endonucleases into the cell to correct them. And people are doing that in every possible way. So in order to put the enzymes, these endonucleases, they don't belong in the cell. These are foreign enzymes for the cell. So they're either using viral vectors or plasmid, or just transferring the RNA itself, or even the proteins. So some people are using charging the proteins, or they have different machinery to deliver these specific endonucleases into. And then if you want to add your own template, then you need some viral vector. Insert your template into the cell. And again, we're using vector because that's what they know to do the best, affect cells. And viral vector, we can look at them as engineered viral particles. We took out or removed the genome of the virus, which makes it replication incompetent, because you don't want to replicate. You don't want to infect the replicating virus, which later will kill your cell. And when you remove the viral DNA, viral genome, you can also space to put in your own sequence of nucleic acids. So there are different vectors. People think lentivirus. It's an HIV-based strain RNA, and that goes back to I heard 2 years ago about viruses. You can use adenovirus, which are linear in the DNA, and people are using more and more AAV. These are recombinant associated viruses, which are linear single-stranded DNAs. Why do people like AAV, if you hear the name in general? Because they are dependent, which means they depend on other viruses to replicate themselves. By themselves, they can't replicate in the cell, so they need the help of another virus, usually adenovirus or herpes virus, to replicate themselves. So, safety point. The wild type is not pathogenic, so it doesn't exist by itself. It's incorporated into chromosome 19, and you can find it in chromosome 19 of many people, but it's considered to be non-pathogenic. Statement was taken from a review May 2015. As for 2 weeks ago, it was found in several patients with hepatoma cells. So I don't know if we can say it anymore. So it might be associated with some malignancy, but so far it's still considered a safe virus. By itself, it prefers HR repair, so it tends to make homologous recombination switching. That's the way it incorporates its DNA. It doesn't use a lot of the DNA or to integrate it within the DNA. It just has homology arms which are similar to sites within the DNA, and that's how it gets incorporated. So by itself, it's using the HR mechanism. Usually it can induce long-term gene expression. It can infect both dividing and non-dividing cells. So that should be the perfect vector. That's a sample of the vector. So in the wild-type vector, you get two sets of genes. One of them are responsible for the replication, and the other is responsible for making the viral envelope. And when you cut those out and you insert your own gene, in this case is an example for how to repair hemophilia B with the factor 9, then you can have your own template. And then there are different types of the virus, so you can select your own types. And each type has tropism to a different organ. So ideally, you can make your template, you can wrap it in a specific AAV and inject it to the body, and hopefully it will target any organ that you're interested in. That's the ideal goal. I don't know if it happens, actually. And this is a way to summarize what we explained so far. So first you make the DNA break. If you're using the NHEJ mechanism, you can knock out gene. If you break the DNA in 2 points, you can create a deletion because you just cut out a piece of the DNA. You can even make or induce translocation if you're interested between 2 chromosomes for some reason. I think this is the part that we are more interested in, and these are the HR-mediated repair mechanism, where you can actually break the DNA, insert your template, and perform gene correction. Yeah.
Does each cell have its own hierarchy of how it wants to repair itself, or is that depending on how you split the DNA?
I think it depends on the cell cycle. So if the cell is dividing and it doesn't have a template, it will use the NHEJ. If it is replicating and it has a sister chromatid or a template, then it will use a template. I think so. Not all the systems are activated in each step of the cell cycle, but still you can get that's a good point because it makes some of the gene editing problematic because you need to activate the cells if you want to insert your own genes. So you need to activate the cell, replicate them. And if you think about stem cells, that's going to be a problem because if they are active and proliferate, they're not going to keep their pluripotency and start differentiating. So that could be a problem. So far, we know how to induce site-specific DNA break. We know how the cell is going to repair it. And we know how to insert our DNA templates so we can do gene editing, which is fine. But we still have another 15 minutes. So I want to give you a few examples and what we are doing now. So CD40 ligand deficiency, and here is the PIDD. It's not a new PIDD, so we should know about it. Obviously, with the adaptive immune system, we want to make a broader repertoire of receptors, either TCR or BCR. B cells are developing first in the bone marrow, then they're going to the periphery, and then continue to mature in the spleen or secondary lymphoid organ. And once they see their antigen, they're going through several steps of class-switching recombination, which means instead of IgM, they're becoming either IgG, IgA, or IgE. And they're also undergoing somatic hypermutation. These are the same breaks which randomly insert nucleotides or remove nucleotides, and you make the antibody more specific with higher affinity, or you make a new repertoire. And how does the B cell know that he has to class switch? The T cell tells him what to do through expression of CD40 ligand. So once the T cell are being activated, they are expressing CD40 ligand, and then there is a CD40 ligand interaction on the T cell and the B cell, and it's CD40, so it's a B-cell T-cell interaction which tells the B-cell that he needs to class switch. These are activated CD4 cells, and you can see that once they're activated, the blue is the nucleus, and the red, I believe it's an immune synapse-like formation. And I stain those for CD40 ligand, so you can see once it's activated, all the CD40 ligand is focused in one place, probably also to activate other B-cells around it. What happens if you don't have CD40 ligand? Then you get hyper-IgM or X-linked hyper-IgM syndrome. And patients have recurrent infections. They have low IgG, low IgAs, low IgEs. They might have either normal or elevated IgM levels. They don't have memory B cell, and they don't have class switch memory B cell because they can't class switch because they can't get the signal. So it looks like a B cell problem, but it's a T cell disease. So why won't we fix the T cells? If we'll fix the T cells, everything is going to be okay. How do we test for CD40 ligand in the lab? Whenever you check this box in the IDL box, test for CD40 ligand, what we do is we take cells, we activate them, and then we stain them for CD40 ligand. These are resting cells, activated cells, and in red, in resting cells, you don't see any expression of CD40 ligand. In blue, once you activate them, then they upregulate CD40 ligand, and you know that the cell is positive. This is a sample from a patient. This patient actually underwent stem cell transplant, but was rejecting his graft. So you can see that there is a small population of CD40 ligand positive cells, some blue cells, most of them are negative. But we also check for CD40 binding, so we're not only checking if the cells are expressing CD40 ligand, because sometimes you can express abnormal CD40 ligand, and you still cannot bind or cannot interact with B cells. So we have like soluble CD40, it's conjugated to some marker, and we can read these markers. So in the control, when you activate them, you see these blue lines or blue histogram, and these are activated cells which are able to bind CD40. In the patient, you can't see any positive cells. So even though it has some CD40 ligand, it can't bind CD40. Basically, these cells are not functioning. So are we the first to think about it, repairing CD40 ligand? No, we're not. And people try to do it. So they used virus to transfer the CD40 ligand first in mice. And even though we get very much better staining, you can see that repaired cells expressed some CD40 ligand. So they achieved some correction. But there was another problem. These mice developed lymphoproliferative disease. And you can see a lot of dots, which means a lot of cells. And these are the thymuses, a picture of a thymus of one of these mice. Big nodular lesions, these are malignancies, basically. And why it happens? Because CD40 ligand should be, it's not the same problem as I described before with oncogene. CD40 ligand has to be regulated. So you want T cell to provide B cell a signal, but you don't want the signal to be there all the time. Because if you're going to get chronic activation or constant activation of B cells are going to proliferate. You're going to get autoantibodies. You're going to get into troubles. So you don't like chronic or constitutive expression of genes. So our approach was a bit different. So we decided to use the talons and cut the DNA in a specific site. And this is the CD40 ligand gene, so it has 5 exons. Most of the mutations are here, by the way. And we decided to target a part which is upstream to the gene before it started to transcribe. And the basic idea, if you make a break here and you insert your template, for example, so you achieve 2 points. First, you get all the regulatory elements which are still there, and you didn't change anything in the regulation of the gene. So when the cell is activated and it's supposed to express CD40 ligand, it will do that. When the cells are resting and are downregulating CD40 ligand, it will do that because it's not constitutively expressed. And the other thing, as I mentioned before, if you get a normal and abnormal protein and CD40 ligand is a trimer, sometimes you can get into trouble. So if you insert it in and you have like a stop codon at the end of your sequence, then nothing of this downstream signal is going to be made. So you won't make the abnormal protein. So you put it in the right site, it's being regulated normally, and you don't get any of the abnormal gene or protein. You should be safe. This is just to show that we're able to knock out actually the gene. So in this case, we use a template, just a green marker. So there's nothing in there, just a green marker. And you can see that if you take cells and you infect them, you get some green cells. And these green cells, when you activate them, you don't get any CD40 ligand, which should be here, which means that we're able to knock out the normal gene, the normal protein. So the next step was, instead of just making the break and putting in a green marker, let's deliver the coding sequence. So we're going to provide the cell. We don't want the cell to work too hard. We are going to provide the coding sequence, which means if this part is going to be transcribed, and when it will be transcribed, it's going to make the RNA immediately that you need to make the protein. No splicing event will be needed. just make it easy. We'll provide you the template. All you have to do, CD4, is make the protein. So we make a template with the coding sequencing of the CD40 ligand. And now you can see that if you check or activate green cells, which means cells that were targeted because we still have the green marker, now they are expressing CD40 ligand when you activate them. And they're not only expressing CD40 ligand, they're also binding CD40 Ig. So this is a functional protein. Can we do the same with patient cells? So yes, we can. We tried and checked different patient cells. This is patient number 2. This is patient number 3. We did the same. We repaired the cells. We activate them. You activate them, you get CD40 ligand. These are positive cells. Green cells, just to mark as a marker that we targeted the cell. CD40 ligand is on the cell surface. 2 different patients, CD40 ligand expression.
You're doing this in vitro. You're not infusing this in the patient.
Not yet, but soon enough, hopefully. Are they normally regulated? So there is always a concern that you're going to activate the cell too much. So you want to compare it to healthy control. So we take the cell, we activate them, and we check for CD40 ligand over time. So we take them at half an hour, 2 hours, up to 54 hours, stain them at a different time point, and you can see that it's been upregulated and then downregulated, both in patient cells, repaired patient cells, and healthy control cells. So it should be safe. You don't get overexpression of the CD40 ligand, and it goes up and goes down as you want. Do they really work? That's the question. So we want to know if they can do what they're supposed to do. And what they're supposed to do, what CD40 ligand is supposed to do, is help B cells. So we had to develop a new assay, and this assay was meant to convince B cell to class switch. So we take naive B cells, they are IgM positive, IgG negative, and we mix them with different cytokines. We leave them for 5 days, and then we stain them to see if they made IgG. And we know now that you can take IL-21, TLR9 agonist, which is an innate molecule for some reason. So I don't know if there is an innate and adaptive immune system anymore. I heard somebody call it the native immune system. And you also add soluble CD40 ligand because you need the soluble CD40 ligand. And when you do that, you can get some up to 10% class switching. So you can convince the cells to make IgGs. Can you do the same with activated CD4s? So in the previous slide, we used IL-21, TLR9, and soluble CD40 ligand. You need the soluble CD40 ligand. We want to use the same mixture, but without the soluble CD40 ligand. We want the T cell to provide the CD40 ligand signal. So we take healthy control cells, CD4s, we activate them. They are supposed to express CD40 ligand. We know that they express it because we check it. And then when you incubate the same naive B cells with activated CD4, you again get this IgG class switching. So we know that they are able to replace the soluble CD40 ligand signal. And the next step is to take patient cells. One of them is missing. We have it now, this data. But you take patient cells, you do the same thing, you repair them, you activate them, and you incubate them with naive B cells. And again, you get this IgG class switching. So if they are mock edited, you get some background. If you edit the cells, you can get similar rates of IgG class switching. So again, yes, we can. We checked this in 5 different patients. Interestingly enough, patient number 1 is the first patient that was described here, actually, with a CD40 ligand mutation. That's a Cell paper from 1993, and we are still following this patient in clinic. We are able to repair his cells. So I think it will be a nice closure at the end that this disease started here in a way, and here we're going to provide some therapy. We didn't end with that. We also you need to abuse some mice in any study. So we took the cells, we transferred them into NSG, SCID mice. We waited several weeks. We took the cells back, which means that we sacrificed the mice. We isolated splenocyte, bone marrow cells, and lymph nodes, and we're able to first see that our cells are still there, so they survive in vivo. And even after you take them out, you can still activate them and they can still express CD40 ligand. So they are fully functioning and they are able to survive. So the CD40 ligand repair basically insert the wild type coding sequence to CD40 ligand site. It results in expression of CD40 ligand and binding of CD40 Ig. It is normally regulated, so it gets upregulated and downregulated when the cells are activated or resting. And we are able to induce B cell class switching, at least in the 96 well plate, So we think that we are ready for T-cell therapy clinical trial. We can stop here since we have 5 more minutes. Just 2 points. It's not the only thing that we're working on. We're making T-Rex, super T-Rex, for example. Yeah.
When you have the chain switching with specific treatment, does it chain switch just one time, or as long as the CD40 ligand is present, do the cells just keep chain switching all the way through?
I think it depends on the cytokine that you present to the B cells. I think the B cells need. You can ask yourself why a B cell decide to make an IgG, not an IgA, and not an IgE, for example. So I think you need the CD40 ligand signal, and then different cytokine environment, and that tells the B cell what kind of class switching it has to do. So if you get more IL-4, then you'll make IgE, for example. I think IL-10 and IL-4 together, I think it drives them to make IgA, but a different cytokine combination which we can use to convince the cell to class switch. Now, the class switching by itself means that part of the DNA was cut, and the heavy chains are arranged one after the other. So it can go further class switching theoretically from IgG, I think, to IgA, and finally to IgE. If it was already chained for IgE, it doesn't have all these IgG or IgA genome in it, so it can't class switch back. So it's being cut one at a time. Yeah. Does that answer your question?
Yeah, I just want to know if the same cell, if you can show it going all the way through by just the continued presence of CD40 ligand, or if you have to change the environment with a different interval.
I didn't try it. I think we can try to take the IgG positive and then incubate them with different cytokines and see what happens. Yeah.
When you use like a retrovirus to insert the DNA or whatever you want to do, what % of cells in the body are affected?
So we do it outside. It wasn't done in vivo. So you take the CD34s. In some cases, you actually condition the patient before you add them back. So you add that outside of the body, and then you either select them. So GFP is one marker. You can't use it in humans because it's immunogenic and it's going to be rejected. But you have other ways to select your cells. Or sometimes they have survival advantage, and then you insert them back. So just by using this is an example. So if you just present template without talents, for example, this is a different one. This is a AAV. You hardly get any green positive cells, so you don't get a lot of cell infected. Once you add the cut, when you break the DNA, then you can get like 30%, up to 50%. It depends on the conditioning, and people are working on improving the conditioning to increase the hit rate.
But once you infuse it in the body, and assuming that you're going to do this, then is it just going to be those cells you infuse, or is it going to go to the rest of the body?
So the virus by itself doesn't have any replication elements in it, so it's supposed to stay in the cell itself. So now with the AAV, you can ask yourself, okay, I might have some AAV. It's a dependent virus. When the patient will be infected with some other viruses, will it start replicating itself? So there are safety mechanisms to prevent that, so it's not supposed to do that. And especially if you have this homology, when you do this homology recombination, it means that you, on both sides of the sequence that you want to insert, you have homology sequence, homologic to part of the DNA. So basically you're going to replace this part, for example, with this part, and you don't going to, and the rest of the viral element will be gone with proliferative division of the cells. So they're not supposed to stay there. And people are looking for that. So one of the things that you do when you study that is that you amplify viral elements and you look for them. Usually we don't find them.
Why did you insert just the CD40 exon?
We thought it would be easier to do that. And there is always problems of the length of, so virus is a very small structure, and there is a limit to the length of the DNA that you can, or the sequence that you can insert. So usually inside an AAV, there is 4.5K length that you can add to that. If it's too long, then you can't do it. So you need, we're already using like, I think 1,000 base pair of homology on both sides, which leaves you not a lot of space. So there is a size limit that you can add.
Do you worry that will affect regulation?
So I think if you think about, FOXP3, for example, that's going to be a problem because there are CNS domains, intronics, regulatory elements. So it should be gene-specific. So in the case of FOXP3, for example, if you want to do it, if you want to replace that, you will probably need to insert intron 1, intron, it's -1, so it's 1 and 2, and then you can continue with the CDS or whatever you want. So it's gene-specific. You can't just do it for everything. Yeah.
Since these patients have never expressed CD40,
they might reject that. That's always a question. That's also, but the same with bone marrow transplant. If you do stem cell transplants, we don't know that. That's a good thing. We'll have to wait and see. So the question is, you basically you present a new. You can say the same for the IL-2 receptor, by the way. So any gene any protein that wasn't there before that, you might develop an immune reaction against that. So they might be rejected over time. We don't know that.
So what's the conditioning stuff that you're going to be doing?
Still to be so. Yeah, we have, off the record, we have 2 patients waiting now in England. We don't know about the conditioning yet. So you'll need some conditioning, probably not biologic treatment to knock out a T cell because you don't want the antibody to stay there when you add back the T cells. Probably some myeloreductive or mild conditioning just to allow better proliferation of the T cells, but we don't know yet.
You've talked about different ways to add things. Has anybody used this in treatment, like with a genetic malignancy where you splice out the genetic abnormality to correct the malignancy? Because you have all these cool enzymes that can cut things exactly where you want. So you think you could slice out the chromosome abnormality that's causing malignancy.
But then you have to target each and every cell. So you need to target all the malignant cells and then cut it. I don't know if people try. I'm sure people are working with that.
In which case you could take it out and then clonally expand.
But you still have the other malignant cells inside your body. So you need to target each malignant cell to do that, I would guess. I think the knockout idea, I mentioned the hyper IgE before. I think that could be used to target the abnormal protein in that case. Just knock out the abnormal STAT3. You have a dimer of STAT3, knock out the abnormal. So you don't know which one you're going to hit. If you hit the normal copy, then you're going to get 2 abnormal copies and the cell will die because there is no living creature without STAT3. And if you hit the abnormal protein, then you get only 1 copy expressed, only the normal protein, and you're doing fine. So we're thinking about targeting dominant negative diseases with that approach. Ex vivo. So you can play with that. So we're trying to make super Tregs and also make them like cytokine bombs. Yeah, if you can make like super Treg, pressing a lot of IL-10 or TGF-beta, then you can suppress everything. That's another thought that we had with WHIM syndrome. With WHIM syndrome, it's a gain of function with the CXCR4, for example. So cells stuck in the bone marrow. And think about it, if you can use the same abnormal gene, put it into CD34 cells, maybe you can perform stem cell transplant without any conditioning just because they have very high affinity to the stem cell, to the bone marrow niche. That's the other way around. And I think the problem is always how you're going to get rid of the gene you inserted in CD34 cells. If you just put it in the CD34 loci, once the cells start to differentiate, they by themselves will downregulate CD34, and the cells will be free to go. So that's something we're trying to test and play with. And third, thank you. I think it's definitely not a one-person job. I was doing most of the CD40 ligand class switching assay. There is a huge group doing that. The huge rolling labs, Andy Scharnberg lab, Troy lab, and Nick. Probably most of you heard the name many times. Nick is an amazing guy. He's a cell editor. You tell him to do something or give him an idea. You don't have to tell him because he does it by himself and brings the idea. And the next day or two, you get a vector with the plasmid or any sequence that you want. We talked once about Zoller and anti-IgE, and he already has a vector with the anti-IgE. We're just waiting for the patent to expire, and then we can infect cells or inject muscles with the IgE vector, and you can get autosecretion of Zoller, and you won't have to get it anytime or more than once. So there's a huge group of people working on that, and I think it looks okay, all promising. No, the sequence is other. So we just copy the exact sequence of the anti-IgE. I think another way we talked about it, just make a vaccine, make the peptide against which the IgE is directed, give a few shots, and then the body will make the antibody by itself. So thank you.
Well, thank you, and you made it very understandable. I knew what you were talking about.
10%. That's what
Raven, did we get a good recording?
Yes.
Things that we were paired to do. Apparently they have introduced a problem